Hello, Pima Aztecs. This is the third video for Chapter 6, Microbial Nutrition and Growth 3. In this video, we're going to look at the media, the different types of media that microbes can grow in. So the media that they grow in is their source of nutrients, their source of food. And we've been talking about this. All microbes need an energy source. They need water, appropriate oxygen levels, because remember, a molecular oxygen can be toxic to anaerobic microbes. They need appropriate environmental conditions. For example, they need to have a certain pH. Some microbes are not fussy about the nutrients that they get, so they can grow in a lot of different media. Other microbes are very fastidious. That means that they need the exact thing that they need, and if they don't get that, then they won't grow. Some organisms cannot be grown in any artificial media that we've made. So for example, the bacterium Treponema pallidum that causes syphilis. You can't grow it in any artificial media, but you can grow it in an animal. The majority of prokaryotes on Earth have never been grown in any culture medium. There's hundreds of types of media available. We'll talk about a few. If solid media is needed to support the growth, then you could put agar, which is a polysaccharide, many sugars, is added to make the medium solid. But remember, agar is not a nutrient. It's just it looks like it could be because it's a polysaccharide, it's a sugar, but it's not a nutrient. It just helps make the media solid. So here in this photograph, this person has made some, mix some agar with the nutrient that the microbe that the scientist wants to grow will grow in, and then pour it into this plate or the petri dish, and then the agar will get solid and the microbe can grow on a solid support. Agar's useful properties: most microbes can't digest agar. It solidifies around 40 degrees C. So temperature sensitive organisms can be added without damage to those organisms, and nutrients, temperature sensitive nutrients can be added and they don't get damaged. So you can use them to grow thermophiles as well, the agar. But there's 6 types of general culture media, and then with each one of those 6 types there's subtypes, and then within those subtypes there's subtypes. In those subtypes there's subtypes, etc. So here's the 6 types of very broad, very general culture media. You could be a defined media, complex media, a selective media, differential media, an anaerobic media, or a transport media, and we'll go over those in this video. So defined media, number 1. A defined media is a synthetic medium. The exact chemical composition for that microbe that you're trying to grow is known. So if you're trying to grow E. coli, for example, here's what they love. You make 1 liter, so you're going to use water, you're going to make a liter of this medium or media, and you're going to put 1 gram of glucose in it. You're going to put 16.4 grams of sodium hydrophosphate in it, etc. down the line. And that is what E. coli likes to grow in. That's a defined media. It's defined. You can see the ingredients are defined. The second type of medium is a complex medium. Complex media, you can grow a large variety of microbes in it. The exact chemical composition is unknown. So you could add blood. So you could take a complex media. You could add blood to the complex media to get growth factors. Then that complex media will be called an enriched media. And here's an example. So you take a rectal specimen and you're testing for Neisseria gonorrhea, and you swab the plate with the rectal specimen. And you can see that you get this overgrowth. So see all these little colonies, these little dots, those are all colonies of microbes. But if you take that same, or a swab from that same sample, you can see that, and you grow it in this Thayer-Martin medium, then you only grow Neisseria. And you kind of have to look pretty close, but you can see them, the little dots here, those are colonies of Neisseria. Chemoheterotrophs are usually grown on complex media, so we come down here in our chart for chemoheterotrophs. Most bacteria are chemoheterotrophs, and you grow them on complex media. The nutrients in the complex media are derived from partial digestion of yeast, beef, soy, or milk. So you get partially, for example, you get partially digested beef and you add it to your medium and you have complex media, and bacteria like that. Here's another complex medium, TSA or triticase soy agar, that grows E. coli, P. aeruginosa, B. subtilis, and Staph aureus. And here is a photograph. So A in this plate, so this quadrant is triticase soy agar, and it grows E. coli. So there's E. coli. It also grows Pseudomonas aeruginosa. B is BHI, brain heart infusion agar, which also grows E. coli and Pseudomonas. Letter C is McConkey agar, grows Pseudomonas and E. coli. And then this bile esculin agar will also grow E. coli and Pseudomonas. And then this is a photograph of nutrient broth that grows Bacillus subtilis, E. coli, and Staph aureus. And for this photograph, these are Bacillus subtilis colonies. McConkey agar, selective medium, will grow gram-negative bacteria. So for example, E. coli and Enterobacter aerogenes. And then after you grow them on the McConkey agar, you can do a biochemical test to look for lactose fermentation to see if these microbes do lactose fermentation. And when we're in the actual lab, we have a lab exam at the end of the semester, and you do a gram stain, and if you have a gram-negative rod, the next test you do, this biochemical test, is lactose fermentation for acid and gas. So this is lactose fermentation is used in these biochemical methods to identify the bacteria. So for example, you do gram stain, you get a gram-negative rod, you do lactose fermentation for acid and gas. If that test is positive, the next test you do is hydrogen sulfide test. If that test is positive, then your unknown microbe that you have is Citrobacter frundii. If your lactose fermentation test is negative, the next test you do is a methyl red test. If it's positive, the next test you do is a urea analysis. If the urea analysis is negative, then your unknown microbe is Serratia liquefaciens. You can also identify microbes using antibodies, but that's an immunological method to ID microbes. And that's actually what they're doing with the coronavirus. They're looking in your blood for antibodies against coronavirus. This is just a test you can do to see if your microbe ferments carbohydrates like lactose or sucrose or glucose. And then you can also test for gas production. So you have these Durham tubes here in each one of these. So what you do is you have your media in here, you put your microbe in, you incubate it, and if, well, the media is red to start with. If your media stays red and there's no gas in these tubes, then you are negative for gas production, and you are negative, your microbe is negative for lactose fermentation, for example. If the media turns yellow, your microbe's in here, media turns yellow, you're positive for carbohydrate fermentation. So in this lactose fermentation test, you'd be over here, you're positive for lactose fermentation because you turned yellow. And then also here in these Durham tubes, can you see this is clear here? That means there's a gas bubble in there. So your microbe not only fermented lactose, but it also produced a gas along with that. This is just what the gram-positive flow chart looks like. So the previous one was a gram-negative. Dichotomous key is what they call them. So if you get your unknown and you do gram stain, you get a gram-positive test, then you look at your gram-positive test and is your microbe a rod or a coccus? If it's a coccus, you do the catalase test. So we've talked about that. And if the catalase test is positive, then you do nitrate reduction. If your nitrate reduction test is negative, you do a Voges Proskauer. If that test is negative, your microbe is Micrococcus luteus. Okay, so let's look at selective media. Selective media contains substances that either positively select for or negatively select for. So if you are a selective media and you are positively selecting for a microbe of interest, then it will grow on that selective media. If you are a selective media and you are negatively selecting for not the growth of a particular microbe, then it won't grow in that media. So, for example, this photograph down here, both plates, they have different selective media, but they were inoculated with the same sample. So it's a sample, contains both bacteria and fungi, but it's different selective media. This plate had a selective media in agar that selected for bacteria. So these are all bacterial colonies on this plate. This was a selective media that selected for fungi. And so you get these giant fungal colonies. You can add things to inhibit the growth of certain bacteria like dyes or salts. This is really important to know. PEA media grows gram-positive and DLA grows gram-negative bacteria. So one way you can remember this is P is positive. PEA grows gram-positive, and then you just remember DLA grows gram-negative bacteria. If you put a lot of salt in your media, that's going to select for a halophile and salt-tolerant bacteria. If you use a acidic media, that would mean a low pH media, then you can positively select for fungi. Okay, fourth type is differential media. So using differential media, and the technique, when you use differential media, the technique is called differentiation. You can see visible changes in the medium. So this looks different than this or this. That's visible change in the media depending on what you are growing. So you're differentiating the organism grown on this one media, on this one plate. So you can use blood agar as a differential medium. And then you can grow, for example, Streptococcus pyogenes here, Streptococcus pneumoniae here, and Enterococcus fecalis on this part of the plate. These different bacteria utilize components that are in the media in different ways. So there's blood. So there's red blood cells, it's RBCs in the media. Streptococcus pyogenes will completely digest the red blood cells and it'll look like this. So this is all where you streaked the Streptococcus pyogenes. And that's called beta hemolysis. Streptococcus pneumoniae does partial digestion of red blood cells, and then it will look like this on the blood agar plate. And that's called alpha hemolysis. And then Enterococcus fecalis does not digest red blood cells. And that's called gamma hemolysis. Okay, and yeah, we just talked about that. More differential medium. So in this photograph, we kind of already went over this. Both tubes contain a sugar, could be lactose, could be glucose, and a pH indicator called phenol red. So if the bacteria do not ferment, and they don't ferment the lactose, let's say you put lactose in here, they don't ferment the lactose, but there's no color change, it stays red. If they do ferment the lactose, then the color changes from red to yellow. That tells you that that microbe growing in there is a lactose fermenter. So A. faecalis on the left did not ferment the sugar, so the phenol red stayed red. And then E. coli, here on the right, did ferment the sugar to an acid and caused the phenol red to turn yellow. Some media can be both selective and differential at the same time. McConkey agar, this type of media, represents both a differential and a selective media. It selects for McConkey agar, will select for gram-negative and will inhibit gram-positive growth. And it will also, McConkey agar will also differentiate between lactose fermenting and non-lactose fermenting gram. So here's a photograph. So in the far left, we have this plate with nutrient agar. You can see the gram-negative E. coli grows in nutrient agar. Gram-positive Staphylococcus aureus will grow in the nutrient agar. Then in McConkey agar, Staph aureus does not grow. So McConkey agar is not letting Staph aureus grow, but McConkey agar is selectively letting E. coli grow on it. And then McConkey agar as a differential medium, McConkey agar will allow E. coli to ferment lactose here, and Salmonella choleraeus, I think that's how you say it, is non-lactose fermenting in McConkey agar. Fifth type of media, anaerobic media. So if you are trying to grow microbes that don't like oxygen, they're anaerobic, oxygen is toxic to them, then you have to use these special chambers that will keep oxygen out of the atmosphere. And then here you can grow your plates with your anaerobic bacteria on them. So obligate anaerobes, you have to have an oxygen-free environment to grow. You can use this stuff called reducing media, like sodium thioglycolate. It will remove oxygen. And the sixth type of media is transport media. You probably, if you guys go on to be doctors or nurses, you will, as hospital personnel, you will have to carry specimens of feces, urine, saliva, sputum, blood, etc. From place to place. You want to do it so that you don't get infected, the sample doesn't get contaminated. Speed is important, but basically this looks like it's a kit to help you transport whatever specimen you're trying to transport. I think that's it.